So the next chapter is really the first part where we really associate biology. We have done a lot of chemistry because we started the atom. We're basically building our next level of complexity, from atoms to molecules, and now molecules, we're going to start building what are known as cells, and we have some building blocks that have to go into cells as well. So we've just a little presentation that goes hand in hand with your notes, so you kind of have them out. I don't have just a picture of your notes, so you just have to kind of take notes along with the pictures that are here, and we'll kind of compare when you get into class how you did. I think it's a skill to definitely work on, is your ability to take notes from just verbal instructions. Some of us are a little weak in that area, so I thought I would try it with this little presentation. So you're seeing here a picture where it says image of cork cells, and that's relevant to this because of the first thing. It's the discovery of the cell. The first person who was credited with identifying cells was the name by a scientist by the name of Robert Hooke, and he's given that credit because he actually looked under a microscope and started observing things, and he looked at cork. Whether you realize this or not, cork is actually the remnants of a plant cell, and when the plant cell kind of goes away, it disintegrates, it dies, some part does remain, and that's actually the cork. And if you recall from a previous chapter, it's actually a polysaccharide that we talked about. It's called cellulose. Cellulose was that polysaccharide that built the structure of a plant cell to keep it rigid. It's one of those also that humans can't digest. So that's just some stuff that relates to things that we've already learned. He called it a cell because if you look at this picture here, to them it was a very simple room, and that's kind of the idea of a cell. Whether you think of it as a jail cell, whether you think of it as like a monk from the old days lived in a very simple one room, and they called it a cell. So he thought a cell was a pretty good name for this. The next scientist there is Anton von Leeuwenhoek, and he took it to the next level. He actually observed pond water. So he's kind of credited with taking it and looking at pond water and seeing that there were other things in there other than just water, but you need the help of a microscope. So we're going to talk about microscope in this chapter as well. The intro to this is really about all the scientists and what they discovered, and we're piecing them all together. So here's the next one. We have 3 scientists that created what is known as the cell theory. So the cell theory is 3 parts. It would be very easy if it basically was just each person gave one part, but it's kind of a collaboration. The first part to the cell theory is that cells are the basic unit of structure and function. So all things that we rely on for the function of our body and for the function of any living thing is really because the basic unit we're built around is the idea of cells. Which then leads itself to number 2, which just says all living things are made of cells. And those cells that we just said are kind of the structure and function to our body. They do everything that we need them to do. And the third thing is that all cells come from pre-existing cells. So you might be thinking, well, where did the first cell come from? And we'll have ideas later on when we talk about evolution later on, and we'll talk about how cells we think change and how they came to be. So for right now, those are the 3 major ideas of the cell theory that were put together by these 3 individuals by the last name of Schwann, Schleiden, and Virchow. Each one has their own little unique thing that they were given credit for. For example, at the left-hand side right here, this is Matthias Schleiden. And what he determined was, by looking at plants, he found that all plants were made of cells. He was very specific in saying plants. He wasn't as general as what the cell theory states, but his partner, Theodor Schwann, you might be guessing, he says plants, this guy comes around and says all animals are made of cells. So together, animals and plants are not the only living things in this planet, but they kind of developed into all living things must have been made of cells or are made of cells. Now, how do you remember that? You can kind of think of it as Schwann, which looks like swan, and swan is an animal, and by process of elimination, Schleiden. Schleiden also is his partner, so plants and animals kind of go together. So that's how, might be an easy way to remember that. The third guy here is Rudolf Virchow, and what he came up with was literally the third aspect that we talked about, where cells come from pre-existing cells. So it's not that each one had the same exact wording as the cell theory, but they took their ideas and came up with what I mentioned right before. So then we have the next section, which is cell diversity, and realizing that not all cells have the same size or shape. In a later slide, I'll show you some examples. You don't have to memorize every single one. We're just going to give you some basic ones to show you that they're not all the same. And we really have to look at this idea of structure and function. So the structure of something often tells you what its function is. So think of something as general as like your hand with fingers. The structure of your hand gives it a good function to grasp things. So that's an idea of where the structure of something can kind of help determine its function. There's over 200 different types of cells in your body. And if you are interested in learning that stuff, that's like anatomy. So you'll learn different types of cells as you go through the different levels of biology. Where most cells are between 10 and 50 micrometers in diameter. So micro being that very small, that 10 to the negative sixth on the metric scale. So this is pretty small, not viewable from the naked eye, but you need a microscope to do that, and that's what we'll work with in this chapter. The next thing are two words that maybe you're familiar with, but here's the idea. They somewhat get mixed together because you can use the same idea of eukaryotic in two different ways. You can describe what is known as a eukaryotic cell. So I can define eukaryotic cell on that first little bullet there, but what that means is it contains membrane-bound organelles. So organelles are these little tiny organs for a cell, and each one has its own function. Just like in your body, you have organs that serve a particular function. Cells have organelles that serve a particular function. And I can identify this as a eukaryotic cell because, for example, you can see a membrane. It's this dark line going around this structure. It's going around this structure. It's going around this structure. Eukaryotic organisms have a membrane-bound organelle. And we're going to talk about that membrane later on. But eukaryotic can also describe an organism. So here is a eukaryotic organism. You might have guessed, what makes up eukaryotic organisms are eukaryotic cells. But the reason why I bring this up is because if ever asked a question about describe differences between eukaryotic cells and prokaryotic cells, students often say, well, eukaryotic cells are multicellular. This is a eukaryotic cell. It is not multicellular. What they're confusing is the fact that eukaryotic organisms are multicellular. So you have to be very careful as to what they're asking you to differentiate between. A eukaryotic organism is composed of millions upon millions of these eukaryotic cells. But this cell is only one cell in size. And that cell has these membrane-bound organelles. When we look at its counterpart, the prokaryotic cell, look at this, it's the same picture because a prokaryotic cell right here first has no membrane-bound organelles. So notice on the inside, there's not even much going on in here, but there's no structures that are going to be surrounded by a membrane. These little blue things, which are identified as ribosomes, there's not enough detail to show this, but they do not have a membrane around them. This is your DNA. DNA, or this organism, this is not you, but this organism's DNA does not get surrounded by a membrane. The only membrane that it is surrounded by is the cell membrane, which it has to differentiate, or it has to be there to separate itself from the outside environment. But there's no organelles inside here that have a membrane. The same picture's up here because a prokaryotic cell is often a prokaryotic organism. Prokaryotic organisms are single-celled. All right, so to go back, to show you, the first bullet for eukaryotic cell is that it contains membrane-bound organelles. The second bullet we can talk about is that eukaryotic organisms are composed of many eukaryotic cells, so they're multicellular. Compare that to prokaryotes. You can say in the first bullet, there are no membrane-bound organelles. And prokaryotic cells make up unicellular prokaryotes. That's what they're called. So instead of calling them a prokaryotic organism, sometimes they simplify it to say this is a prokaryote. All right? So the next slide is just showing you the different types. I had mentioned that there were over 200 different types. I'm not showing you every single type here. But if you look, you can see that there's no two that look identical. Their shapes are different, because all their functions are going to be different. From a brain cell, they have these long axons and dendrites, which you may recognize or know that term, because they send impulses. Whereas other structures help them kind of like a liver. The shape of a liver is going to help you detoxify it. It absorbs things much better. This is not a very good absorbing cell. But these cells that have a lot of surface area, you can kind of see they're more box-like. They have a lot of surface area to them. Blood cells. Blood cells carry things, so they have this concave, and I don't think that this picture gives it justice, but there's kind of a little intratube type thing where you can somewhat put some molecules in there, so they help carry things. So everyone has its specific shape. Like a sperm has a tail to help swim. So there are different things that you can look at right here, and that's just a few. On average, eukaryotic cells are... Sorry, the phone rang. But eukaryotic cells on average are 10 times larger in size. So that's just another difference between eukaryotic and prokaryotic cells. Sorry. All right, the next thing. When it comes to limitations on size, there's two calculations we have to recognize, and that's surface area and volume. From the time that you were born to the time, however old you are right now, your cells haven't just gotten bigger and bigger. That's not how we grow as organisms. Instead, we actually divide our cells. So we're made up of a lot more cells. The size of the cells are the same from when you're an infant to when you're an adult. And the reason why is because of this phenomenon. Surface area and volume, we're going to keep it as simple as possible. We're going to do it into a nice cube because that's an easy equation to remember and still get the same point across. So here are 4 different size cubes, and it tells you in terms of inches, 2 inches, 4 inches, 6 inches, and 8 inches. To figure out surface area, here is the answer, and if you didn't know how to get that, but that's actually the length times width times the number of sides. So 2 times the number of sides on a cube, which is 6, gives us 24. Volume is just length times width times height, and since a cube has equilateral sides, we have 2 times 2 times 2, which is 8. And this ratio is the difference between surface area and volume. So the difference is 3, because 24 divided by 8 is going to be 3. So now we look at something that's larger, this 4-inch cube. Surface area 96, volume 64. So notice what's happening to the surface area and volume. It's decreasing. The larger you get, the ratio is becoming closer. To be efficient in a body, you want to have actually the largest possible surface area to volume ratio, and that's why our cells maintain this smaller size rather than just getting larger. The analogy somewhat thinks about what the purpose of a cell is. You have to be able to get things in and out of a cell. And if you don't have the ability to get things in and out, you're not a very efficient cell. So the volume of the cell you can see is growing at a much quicker rate than the surface area. Surface area is the outermost portion of the cell. So imagine things that have to get in like food and nutrients and things that have to get out such as waste. So the larger you get, the more volume you have, which is all the stuff you're producing, but you can't get it out. So therefore, if we go through cell division, we divide it in half, and then what starts to happen is you create this larger surface area to volume ratio. All right. So that's important, and that actually comes up a lot more even in future chapters. But this is just a little introductory as to why cells, sorry, I moved that. Cells are going to divide more than just grow, grow, grow. So know those two equations, length times width times the number of sides, and volume being length times width times height.